So today he's going to be speaking to us about the promise of precision medicine in alcohol use disorder and PTSD. Charlie, welcome home to UCSF.
Thank you for a beautiful introduction, Dr. Said. Matt, it's a great honor for me to back. I grew up here at UCSF. I have wonderful relations. I was saying to Gina before, when I see my faculty at NYU, I feel very warm to them. I give them a nice warm handshake. When I see my faculty at UCSF, there's no handshakes. There's only hugs because, well, old friends are golden and new friends are silver. So thank you. I'm deeply honored to be welcomed back and to be part of your distinguished lecture series. It's very intense in a kind of nostalgic and sweet and bittersweet way to be back. But it's also wonderful to see the incredible changes in the department. Congratulations on your leadership. UCSF continues to be if not the leading academic department of psychiatry in the country. And your beautiful new home there is amazing. I think it's a flagship institution to represent our field, and I think it's really wonderful. And as someone who spent 30 years at UCSF, I share pride in it with you and wish you great luck as you go forward. And there's so many people I could thank. Many of my friends are here in the room and online, and I had the privilege of working with 3 chairs while I was here. All of them were wonderful and generous to me. Bob Wallerstein and Sam Barundis and Greg Van Dyke, and privilege of working with Diana Nickel, who was my chief of staff at the San Francisco VA. Diana is a distinguished laboratory medicine academic and had a PhD in I think in structural chemistry from Oxford. And actually, Diana was part of the inspiration for my work to try to pursue objective molecular markers for psychiatric disorders. So I owe a great deal to her as well. And there's many, many more, too many more to thank, and all of my friends and mentees. So it's really wonderful. Thank you so much, Matt, for inviting me. So what I'd like to talk today a little bit about is the importance of our field moving from what has been not exactly one size fits all treatment in psychiatry, but treatment guidelines, which are reasonable and somewhat helpful, but not necessarily tremendously helpful for a given patient who's in the consulting room with us, because they're of a general nature. And sadly, in psychiatry, we practice psychiatry the way we practiced oncology in the 50s and 60s. We try a reasonable treatment. If it works, fine. If it doesn't, we move on. But our patients struggle as we try to work through to find the best treatment. So I've been inspired to try to, with others, to try to make the field more like modern oncology or cardiology. These are my disclosures. Primarily receive funding from NIH, DOD, and DARPA now, and some foundations. So the challenges in psychiatry are very well known to everyone in this room and online. We still rely heavily on our patients and their families' reports of their suffering to make diagnoses. And unfortunately, patients frequently underreport their symptoms because of denial and stigma. Psychiatric disorders are highly heterogeneous, like medical disorders, but yet we tend to talk about major depression or bipolar too as though they were unitary concepts. There probably are 4 to 6 subtypes of each of these major disorders, which require different ways to diagnose and treat them. And subtyping is critical for risk course and precision treatment decisions. And our field has paid somewhat less attention to subtyping than has been the case in medicine, for example. I'll talk a little bit about that. So it's like cancer before genomics. Try radiation. If it doesn't work, try surgery. If that doesn't work, try chemotherapy. If that doesn't work, try them all. And meantime, patients are struggling. Practice guidelines I mentioned are helpful, but limited to reasonable trial and error approaches. And first, second, and third line treatments, it must be understood, are based on average responses, not the response of the patient that we're caring for. And we also have limited knowledge about which patients are likely responders to which treatments. I would add which patients are in a given clinical trial. Psychiatry is famous for randomized controlled trials in which drug does not, or treatment does not separate much from placebo. Often they don't get published. Tom Nyland and I actually did a study, I think it was guanfacine for PTSD, and it was completely negative, but we published it in the American Journal of Psychiatry, thinking it's important. Matt Friedman from the National Center for PTSD and I actually did one of the FDA registration trials for sertraline for PTSD. There were 4 trials. 2 of them were positive. They were both in women primarily sexual assault survivors. The other 2 trials were negative. Our trial in veterans with sertraline was entirely negative with male veterans. So it's very difficult. And yet we know the outcomes within the treatment groups are heterogeneous. How can we identify those that would benefit even if on average treatment does not separate from placebo? And I'll show you an exciting new study where we've done that. And the other thing, biomarkers are very difficult in our field. The number one problem we have in psychiatry with biomarkers is that we're the only branch of medicine that does not biopsy the organ of disease. You can biopsy the brain. Neurologists do biopsy the brain for certain autoimmune disorders. We've done it on rare occasion, and it's been very instructive. But mostly we're looking at blood, urine, CSF, saliva, and they're very complex. They're very far downstream from the molecular pathways that are of concern to us. And they're very complexly determined by a host of factors unrelated to the illness. So that the biomarkers we find for example, it's very well known, even the gene expression and epigenetic marks are organ-specific, tissue-specific, and even cell-specific. And yet we look for them in peripheral blood, which is very difficult and challenging for us. So I'm going to talk about precision medicine and some ways to think about precision diagnostics that go beyond self-report, subtyping, personalizing based on molecular circuit and clinical features, and some advanced computational models. And the principle is I remember Warren Buffett was once asked, how are you so successful? What is your formula for being such a successful investor? And he said, I can tell you my formula. It's easy to say and very difficult to do. And it's easy to say what precision medicine is. It's identifying, decoding the heterogeneity of a disorder, in this case, psychiatric disorders, into subtypes that would be diagnostically different and respond to different treatments. Easy to say, extremely difficult to do. All right, so I'm going to present today quickly 6 highlights of 6 studies. 4 of them are published, and 2 are the most sophisticated precision medicine studies we're doing now. And you mentioned that I'm fortunate to be able to continue a large program of research. So we have an NIH center and a DoD center, Dr. Sid, directed at this topic. And I'll just give a little idea about where the future might be. But I wanted to, really my point today in this rounds is to show that at least a proof of concept from several studies we've done that precision medicine is possible and interesting and clinically meaningful in our field. And I'll illustrate that with 4 studies. The first is a study of peripheral blood markers for PTSD, and Owen Walkowitz and Cindy Mellon, Richard Roos, and others have been partners with me in this study. Study 2 is a study funded by the Cohen Veterans Center and the Department of the Defense on voice markers, which I think is exceptionally interesting. The third, I worked with Amil Etkin and his colleagues at Stanford on a study of neural circuitry to advance precision medicine, have some very interesting findings. And the fourth is a computational study with colleagues at NYU, which asks a very simple question. Can we find who are the likely responders in a clinical trial, even when on average a treatment may not separate from placebo? And then two new studies which are in progress, a PL1 looking at likely responders, and to Topiramate for ED and PTSD, and a DoD Senate grant looking at cognitive, molecular, and circuit markers to try the subtype. Okay, study one, and this was published in Molecular Psychiatry several years ago, 2019. It's a very large effort, 13-year effort. Actually started while I was here at UCSF, and I was fortunate to be able to continue that work and continue to work with Victor and Owen, Cindy, and others to continue our work together on this. And the idea in this study was to say most of the previous studies looking for biomarkers, blood biomarkers, for example, for psychiatric disorders, had focused on a narrow range of markers. In the case of PTSD, looking at the HPA axis and looking for cortisol, ACTH, and other features of the HPA axis. But we know that a disorder like PTSD or depression actually involves multiple molecular pathways that are very complexly regulated. And the idea we took was to try to apply a systems biology approach and to really look at genes, gene expression, regulators of gene expression, proteins, metabolites, and look at them in the way that oncologists look at that to try to advance treatment. So the goal, easy to say, very hard to do, was to develop a blood test that might help us in high throughput screening for PTSD in practical real world settings. The use context in this case, because this study was funded by DoD, was primary care or active duty healthcare in the war zone for veterans. And we studied for active duty military as well. We studied male veterans who served in Iraq and Afghanistan. We had, of course, you need to develop these models. You need a discovery sample or a training sample. These are 83 cases and 83 matched controls. And then we had two independent validation test samples. And the most important one was 29 cases and 40 controls, completely newly recruited once we had developed the test from the training sample. So I'll go quickly in the interest of time. We started actually amazingly on these 340, on these subjects, these 82 and 82, we started with 1 million biomarkers. Because when you're doing a multi-omics approach, you have a GWAS, you have the gene expression pathway, so you have a huge number of markers. You can't manage 1 million markers on a couple hundred people. We used a whole series of both machine learning and sort of consensus wisdom of the crowd approaches to reduce it. And we ended up with, we thought, were 343 of the most interesting, unique biomarkers. 2 of these were physiological. One was heart rate. 20 were routine clinical lab measures that you can ascertain in everyday clinical practice. When you go to see your doctor for your annual physical exam, you get these 20 clinical lab tests. We had 8 endocrine markers, including, of course, the HPA markers, 27 metabolites, 156 epigenetic probes. For whatever reason, when we did the complete review of all of the potential biomarkers, epigenetic regulators of gene expression looked to be the most important overall class in developing a blood test for PTSD. Also, small non-coding RNAs, which affect gene expression, 81 microRNAs, 42 proteins, 4 small molecules, and a few linear combinations. So what we found is when we used machine learning, and what we did was we started with the 343 markers. We used a procedure called recursive feature elimination. Basically, this is a machine learning approach in which one at a time you start to eliminate the markers and you watch to see if your classification accuracy degrades. Our overall classification accuracy was high with all 343 markers, and we wanted to see what's the smallest subset of markers that would still preserve the signal to differentiate cases from controls. And in the initial passes, we could, because multi-omic markers are related in the same pathways, they carry some of the same information, and you can eliminate some and still preserve the signal. So that got us down to 77 features from the 343. And we then looked at you get an output from random forest called the variable importance plot, which tells you of those 77 features, which ones are most important to the signal. And so we sorted those, and we found that 28 of the 77 features carried most of the signal. And you know that because as you eliminate them, you get more dramatic drops in the classification accuracy. And what were they? The final panel was heart rate survived, 3 metabolites. Interestingly, lactate and citrate, which are mitochondrial metabolites from the Krebs cycle, they have appeared in every one of our studies. There is something very important about mitochondrial metabolism in traumatic stress, probably in depression also, maybe in bipolar disorder. The mitochondria needs to be a focus of very great careful look. I remember the day in medical school where I finished working on the Krebs cycle, and my thought was, thank God, I'll never have to think about this cycle again. And to punish me for that kind of hubris, Hans Krebs has risen from the grave and said, you'll spend the rest of your life studying my cycle. By the way, of interest, Mike Weiner, who's one of my wonderful colleagues here at UCSF, actually did a fellowship with Hans Krebs when Hans Krebs was a professor at Oxford. 4 microRNAs survived this process, 2 clinical lab measures, insulin and MPV, 11 epigenetic marks, including epigenetic marks important for monoamine pathways, 7 proteins, including prostaglandin. And we found that the final panel had a very good classification accuracy. And what was most important was that this AUC of 0.80, the overall ability of these 28 markers to separate PTSD cases from controls, was independently verified on a completely newly recruited sample. And we had not developed any of these models on that new sample. So it was an independent validation sample. So this is promising, and we are refining these. We think we can do better, pretty well with 11 markers, and we have several ongoing studies to try to further validate these findings. But this is a proof of concept that in principle, it may be possible to use multi-omic markers to do high throughput screening in blood for a disorder like PTSD. Second study, speech markers. I became very interested in speech early in my training. And as I thought about it, human speech is exceptionally interesting. Obviously, we have, I had the following thought. If I call a family member, and within the first 5 seconds of a phone call, I usually know how they're feeling. If I call a patient that I know well, within 10 seconds, I know if they're stable or unstable. And that's a phone call. So there must be a huge amount of information contained simply in the human voice. And I believe the reason for that is that speech is the primary form of human communication. Before the internet, given that we were herd animals, we survived by verbal signaling to each other. So it's evolutionarily developed. With cell phones, speech is simple, non-invasive, low cost, ubiquitous, and it could be used for high throughput screening. So I got very lucky. You have to be very lucky in research. I got a call from a colleague at SRI, just down the road in Menlo Park, who had DARPA and DOD funding to study speech biomarkers in stress and depression. And she said, would you like to, Dimitri Vervi asked me if I'd like to collaborate. I said, tell me a little bit about your group. Who are you? I said, well, we're 25 bioengineers. I said, what, could you tell me briefly some things you've accomplished? Well, we developed Siri for Apple. And we also wrote the code for Dragon Naturally Speaking. So I thought, okay, all right, let's write I said, Dimitri, let's write a grant today. That was enough for me. And this is a fantastic group. And what they do, they study the biophysics of speech. This is not the content of speech. This is not natural language processing. This is the physical wave form of speech. And we studied this in 52 PTSD cases and 77 healthy controls, Iraq and Afghanistan veterans, everyone combat exposed. All of our healthy controls are combat exposed. In our studies, the average duration of speech we looked at was 35 minutes. You can actually do this in 4 minutes, we found now. So the signal is present in very short segments of speech, and they don't even have to be. We asked them, tell us about a neutral event and tell us about the worst, most difficult thing you experienced in the war zone. We find the signal may be there just in speech generally, may not have to do that. So here's what we found. We first isolated the speech segments of the participant from the interviewer. And then we partitioned the way they had a pipeline developed at SRI for some of their earlier projects, like for Siri, in which they can partition the human speech waveform using essentially a form of Fourier analysis into 40,000 unique biophysical elements of the human speech spectrum. And we then took those 40,526 features, ran them through machine learning programs, random forest primarily, and to identify the ones that best differentiated cases from controls using internal validation procedures. We got a classifier with 18 features of the 40,000, and it gave a classification accuracy of 89% to differentiate PTSD cases from controls based on the biophysics of speech with no information about the content of speech or any other clinical variable. We just got a large DARPA grant to replicate this work. If it works, this can be baked into video therapy, into remote treatments, into telephone psychotherapy. And it was interesting to look at the most important features. I don't know if you can read these, but this is typical of the 40,000 features. Peak of deep TV, peak median relative duration. These are, you have to be a PhD in biophysics to appreciate what these are. Validity of coma filters, relative peak location. This is hardcore engineering, right? But these features discriminated cases from controls. And interestingly, I thought they would be features reflecting anxiety and arousal, but they were primarily features that have been shown to be associated with flat atonal speech, muffled speech, and kind of speech that doesn't show the same plasticity and variability in emotional range. And so now remember, these were chronic PTSD cases, 5 to 8 years after combat exposure. Might be different in the first weeks after trauma, but what we're picking up actually is kind of numbing and flattening of emotion as opposed to anxious arousal. Okay, next proof of concept. The question is, can we do in psychiatry what's been done so successful in cancer? Cancer treatment is now based on genomic markers, and monoclonal antibodies have been developed, and treatment is based on the individual features of the cancer. And somebody with prostate cancer may be treated with a lung cancer drug if that's the right drug for them, because it's based on the unique features. So in my collaboration with Amil, Etkin, and colleagues at Stanford, we asked a very simple question. We know that the most widely disseminated evidence-based treatment for PTSD, given that pharmacotherapy has proven to be only modestly helpful for PTSD, is evidence-based cognitive behavioral therapy based on some version of exposure, which has as its underpinning and rationale an approach to fear extinction and memory reconsolidation based on animal models of fear extinction. So that's the model. But we also know from our care of veterans and even civilians with complex PTSD that a large number of patients given this treatment benefit not at all or only minimally. and struggle. So the question is, could we know who is a good and is not a good candidate for prolonged exposure therapy? And Amit's team and those collaborating with him, we took the following approach. We'd like to see if we can identify subtypes of PTSD based on fMRI cognitive network typology and on neurocognitive testing. And the question really was, we know that from other studies, about 1 in 3 patients, maybe 40% of patients, people with PTSD, have significant cognitive impairment. That's quite high. It's true in depression. It's higher, of course, in psychosis. So, but even among PTSD patients and people in the general population, a significant number have disturbances in attention, concentration, and working memory, which can be assessed with neuropsych testing and can be seen in cognitive circuitry in fMRI. So we looked at, of course, the usual fMRI circuits, default mode network, salience network, the visual spatial network, executive control network. We also looked at dorsal and lateral attention networks. And just to remind you that brain circuits are organized into networks. Networks work effectively when they meet 2 criteria. The networks are segregated from each other, so they're not excessively commingled or overlapping. At the same time, the different networks are not only isolated, but they're integrated in a meaningful way. And there are indices, metrics that have been developed to say how efficiently for any given person from their resting state networks you characterize, let's say, the attentional networks as being optimally segregated and integrated. And here's what we found, which was very interesting. We found that among the network integration, this was true for segregation as well, but for network integration, these are the healthy controls in the study. These were female civilian, mostly assault survivors. But among PTSD cases, there were 2 subtypes, those with relatively intact network integration and those with markedly impaired network integration. And this was then replicated in a group of male veterans showing the exact same pattern. So based on fMRI signals, female civilian trauma survivors and male veterans with PTSD both showed a pattern that there was considerable heterogeneity in the PTSD group, with a sizable proportion of the PTSD cases showing network abnormalities. That was also replicated with neurocognitive testing. And here's what we found. Here's perhaps one of the first examples of precision medicine, at least in the field of PTSD. We found this is a randomized controlled study of prolonged exposure therapy versus waitlist, and we separated that into 2 groups: those with and without intact network integration. And the 2 control groups did not do well, as expected. But what was remarkable was among those receiving, this is jumping around, among those receiving prolonged exposure therapy, those with intact network integration and intact neurocognitive functioning had a very robust response to prolonged exposure therapy, and those with significant cognitive impairment actually did not separate from placebo. So there's an example of the use of a biomarker to inform precision medicine, and perhaps as an explanation why 30 to 40% of patients receiving PTSD patients receiving PE do not benefit much. The last study I'll present, that's a completed study, asks a different question. And the question it asks is there a way to understand and mathematically characterize the heterogeneity inside of participants in clinical trials so that we could have much more useful information from clinical trials? And Tom, if we have the data from our guanfacine study, it's probably on some server somewhere. We should actually apply the mathematical models to this and find out, because for sure 30, 40% of those participants actually did benefit from guanfacine. And the question is, who are they? What are their clinical and molecular features? So I was very fortunate that, because I've been working closely, most of my NIH funding these days is from NIAAA, from my alcohol PTSD center. And NIAAA undertook a very well-designed, very well-conducted, randomized controlled trial of a gabapentin prodrug called gabapentin anacarbol. And it was a 10-site trial. And it was done pristine trial, 338 men and women with moderate to severe alcohol use disorder, treated with GEXR, gabapentin anacarbol extended release, titrated up to an optimal dose versus placebo, and at 10 centers. And the result was enormously disappointing. This was a very expensive trial. It was completed, and the result was that drug did not separate from placebo. However, the conclusion was GEXR, 600 milligrams twice a day, does not separate from placebo on measures of alcohol consumption, reduction in heavy drinking days, alcohol craving, adverse social consequences of alcohol use, symptoms of protracted absence. No signal anywhere in the study. Drug did not separate from placebo on the primary or secondary or tertiary endpoints. We asked a very simple question. Okay, should we just write that trial off? It was published. Or should we look to see if we can partition the heterogeneity of the outcomes in the gabapentin group? So we republished the study as a reanalysis, and we were able to identify likely responders. This is complex mathematically. In the interest of time, I'll go quickly, but I just want to present this as an idea which could have enormous value, not just for future clinical trials, but actually for the huge number of declared failed clinical trials in our field, which we believe are actually not failed. We believe that hidden within them is hugely important clinical information that could tell us how to treat bipolar disorder if we knew how to analyze the data. So I work with, first of all, you have to be careful who you ask to work with you as statisticians. And one thing I stumbled into, again, serendipity, like that call I got from SRI, from the Siri team. I happened to meet two statisticians at NYU, and I noticed that they weren't typical statisticians, that actually they had PhDs in mathematics. And they were PhDs from the Cohen Institute. And so they don't actually do statistics at all. They do statistically informed mathematical learning. That is to say, they don't apply off-the-shelf statistics. They look at the structure of the data and ask what is the right mathematical model to answer the question from first principles. And I'll show you an example of their model. And in this case, it was for likely responders. And it's a complex prediction process using random forest. What we knew that we had from before the patients were randomized in this NIAAA file, we had data on 223 patient characteristics. We didn't have biomarkers data yet, although we have it now and we're working on it. NIAAA has shared the DNA and RNA for all of these subjects with us, and that's for coming attractions. We have I just got a new R1 to do that. But here we had demographics, clinical information, psychiatric diagnoses, clinical features, neurocog testing. We had clinical features. And we asked whether we could use these clinical features, which we were ascertained before randomization, to determine who would and would not respond to gabapentin anacarbol. And this is a complicated approach. The simple idea, if you ignore the slide for a moment, the simple idea is the following. Certain people with a certain set of features are going to be good or bad prognostic cases. The problem is you just don't know which features. But if you have 223 features, you can develop a vector of features for each person and ask whether they respond to the drug. And then what you could do, which is very interesting, is you can match them to people who are randomly assigned to placebo, but share that same vector of features. So in a way, it's a poor person's way of twinning the treatment subjects with their identical feature match in the placebo group. And then if you can identify which features are associated with good prognosis in the treatment group, you can look to see if those people with the same features did or did not do as well in the placebo. So that's what we did. This involves causal modeling. That was developed by Hansen. Interestingly, the brother of Helena Hansen is a distinguished colleague with us at NYU and now at UCLA. And he developed the causal modeling to be able to, so to speak, do this pseudo-randomization where you now have cases and controls that are matched on the prediction features. So you fit them all out using the baseline features. You obtain the probability of response to treatment to GEXR. You find those who will do well in the GEXR group, the subgroup. You match them in the placebo group, and then you compare them. And when we did that, by the way, this is the model, which is why you want to collaborate with mathematicians, not statisticians, because if you collaborate with a statistician, no one can tell you how to do this. They developed how to do this from first principle. And lo and behold, even though if I showed you the curves for the study as a whole, cases and controls were completely on top of each other, no effect. Using this method, we found a subgroup of the cases, which we call likely responders, who clearly separated from a subgroup that we call unlikely responders. And we then determined their characteristics. And here's what we found. Even though the group of people on gabapentin prodrug did not separate as a whole, the patients with higher numbers of heavy drinking days in the 30 days prior to randomization, lower levels of anxiety and depression, and higher levels of cognitive and motor impulsivity on the measures we had, did better and outperformed placebo when they were matched on those vector of characteristics. So gabapentin, which is a mood stabilizing anticonvulsant drug, is really good, it appears, for alcohol use disorders, for people who are more impulsive, more cognitively disinhibited, have more motoric disinhibition, and drink more heavily, and have lower levels of, so to speak, internalizing symptoms, which is, to me, and I'll bet we could do this for each failed major clinical trial and find a subset. And what I like about this, these findings are immediately actionable. You can use them this afternoon with an alcohol use disorder patient you're treating. And so this was the summary of the findings. Okay. And how am I doing for time? I'm okay? Good. I just want to say, so to summarize, I presented 4 proof of concept studies. A blood test that we're trying to validate to say that it may be possible in the future to have high throughput screening for psychiatric disorders, even in a population of veterans who are reluctant, especially active duty, reluctant to tell you they're struggling. And we think, by the way, we can do better if we move from peripheral blood to neuronally derived exosomes, where we might get a better signal of what's happening in the brain. Lynn Pulliam here at UCSF has done that for HIV, neuro-AIDS, and neuro-COVID, and we're going to attempt to do that with Carla Nasca and Betty Dijon at NYU. So I presented the idea that we might develop blood tests to screen for a psychiatric disorder, that voice may be used as a low-cost, ubiquitous, high-throughput screening marker for depression, for PTSD, for other disorders, and that brain circuits might tell us who does and does not respond to psychotherapy or pharmacotherapy, And finally, we can mine clinical trials, even failed clinical trials, to get meaningful clinical predictors of who will respond to treatment. So in the next 5 or 10 minutes, I'll tell you where we're taking this work now. So we're in the middle of an NIAAA funded PL1. And the research question in this trial, I've said already what the problem is in clinical trials. So I'll jump ahead. The model we're using was developed by George Schub to look at Topiramate for treating alcohol use disorder, PTSD, and it's a 3-phase model in which the molecular pathways are different for binge intoxication in the early phases of alcohol use. Then they're different again during the dysphoric withdrawal phase, and then finally different again in the rigid cognitive preoccupation and anticipation phase. And we think that for a whole variety of reasons, Topiramate will be helpful to address this problem. And in particular, in alcohol use disorder, one of the most important dysregulated metabolic pathways is in glutamatergic and GABA signaling with an imbalance of glutamate and GABA signaling leading to network hyperexcitability. Topiramate has been shown in animal models and some human studies to reduce hyperexcitability by restoring this excitatory inhibitory imbalance. And so that's the structure of this. It's very complicated. It has imaging cores and molecular cores and neurocognitive cores. I won't go into all the deep rationale for Topiramate other than to say our work was based in part on Steve Baki's work at UCSF on looking at Topiramate in PTSD and AUD. So this PL1 is a phase 4 double-blind 2-group randomized controlled trial of Topiramate versus placebo in 150 participants randomized 2-to-1 to Topiramate versus placebo. We have to be careful with Topiramate. Tom was the first person that told me that neurologists call Topamax dopamax. I've always remembered, Tom, that you cautioned me about that. There's a lot of cognitive blending with Topiramate. So we dose it up 25 milligrams a week and not faster than that each week for 8 weeks. And we stop when the patient can't tolerate a higher dose, which is usually 100 to 150 milligrams. And doing that, we've successfully been able to manage the patients in the trial. And we're going to do essentially what I want to do in this PL1 is take all the lessons learned from the 4 studies that I presented to you and embed them in a clinical trial. Get all the molecular markers, get all the circuit markers, get the neurocognitive testing, use them all using this mathematical model for determining likely responders, and then have a deep answer to the question, who does and does not benefit from Topiramate if they have comorbid alcohol use disorder and PTSD, and do this as a really serious integrated study. Meantime, we're also collecting voice markers in the study. We're getting video recordings for using Paul Ekman's facial action coding. Patients have an empathical watch, so we get heart rate variability. We want to try to model all of this to try to advance precision medicine. And so this is the overall design. You collect the molecular markers, the circuit markers, the digital markers. You embed them all in the clinical trial. You use advanced analytics, and the output is we really don't even care if Topiramate separates from placebo. What we really care about is what clinicians care about. Does my patient who I'm treating this afternoon, struggling with 20 years of heavy drinking and following childhood trauma, can they benefit from Topiramate, and why Topiramate and not something else? That's the final thing, and I'll stop here and leave time for questions. We just got funded to do a major study. This is a DoD center study, 4-year study. We'll study 5 existing cohorts. These cohorts are already completely ascertained. All the studies have been completed. All the blood has been drawn. All the imaging has been completed. All the diagnostic testing has been done. 3 veteran cohorts, 2 civilian cohorts, and includes the Aurora cohort, the best done study in civilian PTSD that Perry Ressler is collaborating on this with us. And in total, we will have 3,000 PTSD cases and 12,000 controls, all of which have been deeply phenotyped clinically, neurocognitively, molecularly, and from a neurocircuitry perspective. The research question is very simple. Can we partition PTSD into meaningful subtypes? And we're going to try it 2 ways. The first way is I did a very intensive review of the world literature on clinical subtypes of PTSD, such as they are, and we came up with 4. There's several other candidates, but PTSD with impaired cognitive function, milder, less comorbid PTSD, severe, more comorbid, more complex PTSD, and PTSD with pronounced depressive anhedonic symptoms. There's definite evidence in the literature clinically for these subtypes of PTSD. There's some evidence for a dissociative subtype and some others, and we'll look at them in a secondary question. So one way to do this study is to start with these clinical subtypes and then say, can we map the molecular pathways and circuit features onto these subtypes? And for example, these are the 7 major molecular pathways we've identified in our studies, and we have the ability using our multi-omic platforms to ascertain all these and other markers. You'll see the regular subjects, oxidative stress that Cohen's group is working on, the Victor neuroinflammatory pathways, neuropeptides and neurotrophins, monoaminergic pathways, reward, and then the GABA glutamatergic pathways. And neurocircuitry work that we've identified in an article we published in the New England Journal with Israel Librazan and Arik Shalev, we identified 4 specific neurocircuit phenotypes for PTSD. I don't want to go into all the details, but we have an emotion regulation circuit, a fear-leveling circuit, which is the best known amygdala-based circuit for PTSD, a threat detection circuit involving hippocampus and medial and lateral prefrontal cortex, and a contextual processing and update circuit involving hippocampal-dependent processes with insula and PFC. So my point is we have 7 well-established molecular pathways, 4 well-established circuit phenotypes for PTSD, and what we're going to do is do what is not usually done, which most of these studies are usually siloed. So there's a study on circuits, there's a study on molecular pathways, there's a study of clinical features. We're going to try to integrate all this information, and this is the full model. We have the 4 clinical subtypes: impaired cognitive, impaired cognitive type, the milder form, the complex severe form, and the anhedonic form. And we're saying we've pre-specified different molecular pathways and circuit pathways that we think should differentially associate with these 4 subtypes. So that's the first approach we're taking. We're going to flip this on its head and take a second approach in which we start with biotypes rather than clinical subtypes, identify biotypes of PTSD, and see what clinically associates with them. But the goal of that study is to advance precision medicine by saying there's not PTSD, there are multiple PTSDs. They're different. They have a different history. They may have different trauma histories. They may differ by gender, for example. They are different in terms of their molecular pathways and circuit features, and they respond to different treatments. We know that people with the cognitively impaired subtype don't respond well to PE, as one example. So I'll stop. I think I want to just acknowledge the fact that I have a wonderful group of collaborators. The study presents cores. So we have the NYU core, we have the UCSF core with Owen, Cindy Mellon, Victor, and Winnie Wu, and that's a metabolic, primarily metabolism focused core. The Army is a DNA and RNA core for Dietrich. We have the voice marker core at SRI, the endocrine cores at Mount Sinai with Rachel Yehuda and her team. The computational core is based out of Harvard with Frank Doyle, who's the Dean of the School of Engineering at Harvard, Terry Ressler and others. And then we have a proteomics core with Leroy Hood. It's always nice to have Lee Hood on the study. Since he developed PCR, we think we're in good hands to do the genomic work. So I want to again thank all of you, and mostly I want to thank you for the wonderful pleasure and privilege of having been a member of your family for 30 years. I have deep affection and deep gratitude for all of you and for my UCSF family, and whatever modest success I've been able to have at NYU, it's because of my experiences and relationships here. And thank you, Dr. C.
Great. We have about, I don't know. Great. Top.
Story was Andy Miller's infliximab trial in major depression.
Yeah.
The overall trial was negative, but people who had elevated CRP, immune marker, responded to infliximab, and people with low CRP actually did worse than placebo.
Correct.
So, and because it was an immune-targeted intervention and it was the immune biomarker that actually accounted for these subgroup differences, that was like a home run. Which gets at my question. So our drugs have such protean effects on neurobiology, and I'm just wondering whether if we could look at interventions that have more of a discrete focus of engagement, maybe TMS, for example.
Yes.
Or, as opposed to drugs like Topiramate.
I totally agree. So it's a wonderful question. The problem we have in psychiatry, one of the problems we have, is that the mechanisms of our disorders are very complex and involve the integration of molecular and circuit features across the brain. That's one problem. So we don't have a one target. I mean, as much as NIMH would like us to have a target to engage, the target is the brain, unfortunately. And the second problem we have is that our drugs, actually, one of the strange ironies are, the more selective our drugs are, the worse they are. The worst drugs in all of psychiatry are the highly selective serotonin reuptake inhibitors. And anytime you can use venlafaxine or Wellbutrin, you'll do better off than you will in most cases with a highly selective SSRI. So it's a big problem. However, there's a much better chance with TMS. There's a much better chance even with DBS probably, or deep focused ultrasound. And maybe even in psychotherapies, that might be very specific targets. So I do agree with you. But that being said, I think when we have a better understanding of the subtypes, we'll begin to be able to think more clearly about which treatments to pair with them. That will be the next stage of the work. And I'll give one very simple and very sobering example. We treated a patient in my department with 20 years of unremittent treatment refractory depression. And the patient failed all treatments, including ECT and TMS. At some point, neurology did a consult and thought he might have an autoimmune encephalitis. They did a brain biopsy. They found massive neuroinflammation and treated the patient with infliximab, and the patient became improved for the first time in 20 years. So the other thing, Tom, is we do all this with one hand tied behind our back because we don't know what's going on in the brain.
David.
Yeah, really beautiful work, especially based on your final study where you're using the biomarkers to help try to define the different subtypes.
Yeah.
Have you gone back to the first two studies to see where your extra 10 to 20% of sensitivity is? Is it in a failure of the diagnosis? But, do you know why you were only getting 80 to 90% sensitivity? So in the first two studies, you were getting like 80 to 90% sensitivity, of your biomarkers and speech to predict to separate. Have you gone back to look where's that extra 10 to 20%?
Great. So I found it very encouraging. Now we have to replicate that, by the way. Those were in male veterans. They might not replicate in female civilian trauma survivors. But the fact that we could get reasonable classifiers, accuracy, AUCs of 0.80 with heterogeneous groups, we will definitely go back and try to look to see, is the signal coming from, is there a differential signal? Is the accuracy different as a function of the subtypes? Is voice quality different? Let's say we can validate these 4 subtypes for someone. We have to go back and re-examine voice quality. So wonderful suggestion. We should be able to do better. And in particular, we should be able to do better with blood biomarkers, because they're the noisiest features. And I'm hoping, fingers crossed, that neuronally derived exosomes will tell us much more about this molecule.
Wonderful. Thank you so much. You have tons of questions on the internet. We're giving the advantage to people in the room. Those of you on Zoom, mind that. It is. We're trying, but positive reinforcement. Thank you all for coming in the room, and thank you so much for a wonderful talk.